All right. Well, good evening, everybody who's out there. I'm Scott Rodeo from Special Surgery. I'm with my colleague and co-chair of the ASS Shoulder and Elbow Society Research Committee, Peter Simon. Tonight is the third in a series of webinars from the Research Committee, and tonight we're going to focus on orthobiologics and some thoughts about establishing both clinical registries and, importantly, a linked biospecimen repository. Just by way of some brief introduction, I think we're all aware that this whole area of orthobiologics and regenerative medicine is an area that has tremendous potential. Common area we hear about from our patients all the time, but I would submit to you it really needs further development. There's a lot of unanswered questions. I think we've all seen aggressive marketing and sometimes indiscriminate use. And so an area that really has great potential and needs really further development. And probably the single most challenging aspect to this whole area is the heterogeneity in these different formulations. They're all different. My PRP is different than your PRP. And so to that end, clearly we need to study our patients, and not just a clinical registry, but we recognize the importance of having a biospecimen where you can take a small aliquot of what's given to the patient so we can take it to lab and look at composition, cells, proteins, and ultimately identify some markers of biologic activity so we can correlate the composition or biologic activity with clinical outcomes, with imaging outcomes. So I'm joined by three of my colleagues from around the country tonight, J.T. Tokish from Mayo Clinic in Arizona, Shane Shapiro from Mayo Clinic in Florida, and Ken Zaslow from Northwell in New York. All of these individuals have a lot of experience in this area and real expertise to share, and they lead regenerative medicine initiatives of their own institutions. And in fact, we were collaborating on a multi-center initiative. So I'll have some talks tonight on some thoughts about establishing linked clinical and biospecimen repositories. First, and perhaps most importantly, Smith & Nephew has been gracious enough to sponsor this. And so we thank our sponsor, and we have a brief video to show right now.
Good evening, everyone, and welcome to the ASCS Foundation webinar series. I'm Scott Gunn, Vice President of Shoulder Marketing and Sales at Smith & Nephew. As the sole sponsors of this series, we're thrilled to have the opportunity to introduce you to Smith & Nephew and our innovative product portfolio. For over 100 years, Smith & Nephew has been at the forefront of medical technology, committed to improving lives through our pioneering solutions. We're excited to share our latest advancements in shoulder arthroplasty and sports medicine. Our team of experts have been focused on providing you options throughout your patient's continuum of care with cutting-edge products like Regeniten, a bioinductive implant that redefines biologic healing for rotator cuff tendon growth, and the elegant ATOS shoulder system, which embodies stability, simplicity, and flexibility. ATOS is a streamlined system for both anatomic and reverse procedures, allowing for an elevated operating room experience. Throughout this webinar series, you'll hear from leading experts in the field who will discuss the latest research, clinical evidence, and best practices in shoulder and elbow medicine. Thank you for joining today, and we look forward to sharing our passion for innovation and helping you provide the best possible care for your patients.
Thank you. And with that, why don't we jump right in? I think JT is on yet, so we have Dr. Ken Zaslow from Northwell in New York to talk to us about some thoughts about the clinical and laboratory data that we might be able to collect in our patients.
Okay, I think I'm sharing. Does everyone see my screen now?
Yes.
Perfect.
Great. Okay, so we're going to talk a little bit about, as Scott said, how to set up a bioregistry and biorepository. And I'm going to start with a little bit of the more boring stuff. How do you set up the registry? What is the data? They asked me to discuss what data should we be collecting? What are the points of interest for us, both clinically for our patients and research-wise, if we're going to try to figure out what works best and how to do this. And, as Scott said, every one of us has different contribution in our blood and in our fat and other things we might use in bone marrow. So it's important to understand what it is. What is the drug that we're testing? And everybody's is slightly different. It's very simple when you have a drug that you're testing for a randomized controlled trial because you know exactly what that drug is and you're just looking at outcomes. Here, we're not only looking at outcomes, but we're comparing the outcomes I'll discuss with what was in it at the time of injection. So let's start. I can't seem to move my slides. Okay, there we go. So we start right away with some just general information on who you're injecting. The physician mostly this data is de-identified if you're going to be sharing it among groups or entering into a larger registry. But if you're going to be following these patients up yourself, obviously you would need the name, date of birth, and contact information. But on the left side, you see the de-identified information that we need. Who's the physician that's injecting, which joint, the laterality, and then basic demographics. Then some general history questions about what it is that the patient has. Have they come with a diagnosis of either OA or a tendinopathy or tear? Which joint are we dealing with? Obviously, for this group, we're talking about the shoulder. Have you had any procedures done on the shoulder, whether they're injections, other previous injections, or surgery? And if it was injection, what was injected and when? And if surgery, what year and what procedure? And then, of course, you like the general sense of what meds the patient is on. The next set of questions is the patient-reported outcomes, and you have to pick which ones you want as you set up your registry. These are ones that you might consider in a shoulder situation, the VAST, the ASES, probably the most common, then the simple shoulder tests, any of the PROMIS tests, the WOOS or the SPAD, and the constant score. I would suggest that for patient compliance purposes, that is to get patients to regularly fill this out every 3 to 6 months, it's best to try to limit the number that you do and try to avoid too many redundant questions. But 2 to 3 PROMs is very reasonable, and that's what we have in our bioregistry for OA of the knee. Then before the procedure, you need some general information that has to be noted, which side and which joint, what was the pathologic condition, and if there's an x-ray, which there should be, what is either, at least in shoulder and knee, you'd use Kellgren-Lawrence, and then shoulder, you can use Kellgren-Lawrence score or the Samuelson-Prieto score, and any other preoperative imaging such as MRI or CAT scan, if applicable. Then you want to look at the point of delivery. What is the injection that you're giving? Okay, so obviously what product? Is it a PRP? Is it BMAC? Is it lipoaspirate or amnion? Once that becomes available again. What if it's PRP or BMAC? What is the centrifuge that you're using? If it's lipoaspirate, what way are you micronizing the fat? And if you're doing PRP, we need a little more information. Is this leukocyte? Are you preparing leukocyte-rich or leukocyte-poor PRP? Are you adding fibrin? Are you activating it in any other way? And then, of course, you need to know the volume of blood you drew and ultimately what you concentrated down to and the volume that you injected. Lastly, some other things that are important are things, what else did you add to the injectate? For example, in a blood product, both BMAC and blood, you might add anticoagulant. If you did, which anticoagulant, what volume? Did you add an activating agent? And furthermore, was there any other things you treated the patient with concomitantly? For instance, there's evidence that people can use HA plus PRP. Did you use HA? Did you maybe add concentrated platelet poor plasma? Some people not saying that we're recommending, but they're talking about adding Kenlug. But whatever you add at that time has to be listed. Every one of these things can make a difference in your on your PROMs and in your evaluation. You also want to know the time from collection of the blood to injection, the total volume injected, what gauge needle you used to see if that made any difference, and did you use ultrasound guidance? Lastly, if you chose to drain any fluid from the joint, was there synovial fluid collected and what volume was collected? The next set of questions really relates more to research. That is, if you're looking, although a lot of the patients want to know what was the dose, they've read the data, they understand that higher doses do better than lower doses. And so they may want to know this. So it's at least all of us who are in the registry for NEOA that Scott's part of and Shane, we had what's called a Hariba. It's like a countertop cell counter. And you can immediately do a CBC on the whole blood that you drew. And then once you spin the PRP, you can do a CBC on the PRP. And you can get not only the exact concentration of your platelets, but just knowing a little math, you can figure out what was the number of platelets in the billions that you actually injected, which I think is important as we start to look at results. And then, of course, what's the leukocyte? How many leukocytes? And within white cells, how many of them are monocytes and how many lymphocytes? and if there's any RBCs in your white cell in the rich, the white cell rich. Next, we need to know about the biorepository sample. Shane's going to spend more time on how we store these and how we keep them, but at least in your data gathering, you have to listen, was this taken for research? How much was kept? So for instance, if you took a half cc, did you divide it into 4 aliquots before you froze it and then you freeze it? Or, or how many did you keep? So you have a good track for that. Was there a whole blood sample taken, and did you separate that into some aliquots for storage? And was there any synovial fluid? And you would keep that as well. And then what's the degree in the freezer that you're storing this in? And at how long from the time of blood draw and giving the injectate did you actually store this into some freezing, separate it to aliquots and freeze it? Scott asked me to discuss a little bit about MEVO characteristics. So what are MEVO characteristics? Those are the minimal information that's needed for effectively studying, evaluating biologics in orthopedics. A way to make it appropriately that we are comparing apples to apples when we look at various studies in the literature, because without these characteristics, it's very hard to tell. So if the study is of a minimally manipulated tissue like we're doing at the moment, PRP, BMAC, or MFAT, these characteristics may not be necessary because you're not calling them stem cells. But if you are going to start doing true stem cell research where you culture bone marrow or you culture fat or blood that's been affected, and you could culture that for stem cells or what we call medicinal single cells, MSCs, these characteristics should then be reported. And this is a much more detailed reporting than I just gave you in a moment. And I'll just quickly go through those. First of all, what is the stem cell source? And then issues that, what is the tissue storage media and what was the environment it was harvested in and kept? Next, looking at the processing of the stem cells, was there a detailed protocol of what you did to these cells? If you used a commercial system, which one was it? Did you digest it? What solution and concentrations did you use? Was there agitation or some type of cleaning of the tissues? Was there any other type of method of purification? And then what did you end up with at the end of this process? Next, if you're going to cell culture, which of course you would do with stem cells, you need to know all these conditions related to the cell culture and description of the stem cell characteristics, including their immunophenotype and details of their in vitro differentiation. Lastly, how did you deliver the stem cell? Was it in some type of media? Was it concentrated further? Was it given with growth factors? And was it attached to some type of scaffold? And once you do that, you've given a lot of data, and then we can start to compare things apples to apples. But once again, at this moment, for PRP studies, this is probably a little bit overkill. Thanks very much. I'm going to move this on to Shane.
Perfect. Yeah, Shane, why don't you go ahead then?
Okay. All right. So I'm going to share my screen here.
Well, Shane does it. Just for a brief introduction, Shane is primary care sports medicine at Mayo Clinic in Florida and has been studying this area for quite some time and well published in this area.
All right. Does that look okay, everyone?
Perfect.
Yeah, perfect.
All right. So Ken did a nice job there talking about the different things that we can look at. I'm going to take just a couple of minutes here to talk about how we operationalize it all before JT wraps a bow around the big projects that we've got going here. A little bit of a hobby horse before we start on the operationalization. I think we all know, as Ken alluded to, that there's a number of different products out there, and some of these are pharmaceutically manufactured under GMP conditions, whereas some are just same day, same patient, same surgical procedure orthobiologics, and these are held to different standards. So a lot of what we're talking about right now is the simpler same patient, same procedure, like PRP, BMAC, and MFAT. But a lot of the same rigorous research goes into the pharmaceutically manufactured products, not just the ones that we're talking about here tonight in our practices. And The reason for that is conventional drug discovery, drugs defined as substance intended for use in the diagnosis, cure, mitigation, and treatment of human disease. They have to go through this phased stages of development from discovery to preclinical to phased research before they're approved for use in our practices. But as I'm sure we're all aware, in the world of biologics, this is an extremely high cost for the drug development process, upwards of $1 billion on average, and somewhere towards 15 years to get an actual biologic pharmaceutical to the market. And so a lot of what we're using with our orthobiologics utilizes the 510K device approval pathway, and that has been the simplest pathway to utilize orthobiologics in our practice up to this point. But we know many of our surgical procedures have come through innovation in the operating room, and a lot of these procedures that we're talking about tonight are the same way. And so that differs a little bit from conventional drug discovery. But what we're talking about when you talk about biorepositories and registries is how do we bring the two together, traditional surgical innovation and also conventional drug discovery. And so this is a challenge for us because these first generation orthobiologics, we're allowed to use them in practice because it's an off-label use of a 510K cleared device. But then we think of them as procedures, but in reality, in some respects, they're products as well. And so everything that we're describing tonight is a challenge whereby we are the product developers, we're the product utilizers, and we're also tasked with being product evaluators. We're talking about doing this research ourselves, and there's not many instances in medical history where a group of clinicians and scientists have had to do this way. So this is the way that I think of what we're talking about. You have a scientific idea, even if it's something as PRP, then you got to figure out how you make the PRP and the process development. You develop some SOPs and some policies. But then on the way to getting the product to the patient, you have all these other issues like manufacturing, quality control, how do you handle the product, how do you store it, and then what's the complex regulatory oversight and ethical considerations involved with all of that. So all of that is background into how we collect these samples. All of this starts with an informed consent process. If patients are participating in the research that we're conducting, we need to make sure that we have their informed consent. Ken talked about starting the sample collection and the things that we want to collect, and then I'll briefly go over the basics of how we process, store, and bank these. Starting with the informed consent, though, I'd like to point out when operationalizing a project such as this, the clinical research coordinator is the lifeblood of this project. I always think of myself as the front man and the guy on the end of the procedure, but everything in between in terms of communicating with patients, study participants, assisting with the informed consent, scheduling these procedures, scheduling the follow-up, scheduling the patient-reported outcome measures that Ken mentioned, and then possibly even sample transport to the banking institution. All of that has to be coordinated by a solid clinical research coordinator. And so when we think about integrating such a project into our practices, a lot of times we think that a medical assistant, a clinical assistant, a nurse can handle a lot of these procedures, and in many cases they can, but sometimes the sophistication requires someone trained in the research environment who knows how to operationalize all of this. So once you've got your clinical research coordinator on board, and you go about the process of collecting a sample, whether it's PRP, whether it's whole blood, whether it's adipose tissue, whether it's bone marrow, you've got your sample, and you want to have some place where you can collect it, whether it's a tube, a specimen tube, or just a syringe. That'll be something decided at the research onset. The collection process can be as sophisticated as requiring a barcode, and talk about a laboratory information management system as the barcode being part of that. And that's just the way that we can track all of these. And so, you know, it can be as simple as handwriting on a label on the sample collection, or it can be as sophisticated as a barcode. The processing is the preliminary processing involved in the storage. And without going too deep into this, we might ask ourselves when we're studying PRP, do we want to study PRP as the whole sample PRP that we're delivering to the patient? Or do we want to activate the PRP before we store it? And this was something that was discussed at the time the Biologic Association Biorepository project was getting started. Are we storing PRP, or are we storing the biologic, the growth factors that are released from the PRP? And so that's an important decision to make. Once you've processed the samples, they go about the storage process. And that, again, that's a scientific decision for what is the best way to preserve the samples for the purpose of the investigation. Again, entering storage conditions, the storage date, what temperature you're going to store them at, all of that should get entered into a laboratory information management system if you're going to be using one. Again, you can still do studies without a LIMS. You know, a simple red cap or an Excel spreadsheet to log all of this is perfectly acceptable for a simpler project. And then finally, if you do store the samples long-term, a true biorepository is going to take requests from your research collaborators for the purpose of pulling the samples, aliquoting the samples, and shipping the samples so that they can be studied for biomarkers, proteomics, and whatnot. Patient-informed consent is crucial. You can't keep anything with a patient identifier, whether it's a patient-reported outcome or a patient sample, without getting their informed consent, unless it is de-identified. So we do have waste IRB protocols that do not include patient information. So you just give the patient a number, and you can record age and some other demographic variables like that, as long as it doesn't have patient identifier. We call those waste protocols, and you can still do some science that way. Whether or not your collection and your storage is being done by the CRC or by a medical assistant, I mentioned that already. And then you typically would want to have a manual documenting all the operational procedures and make sure that everyone is adequately trained to perform this. Some samples can be stored right there and in a local laboratory, and then some will be sent off to central housing. Again, it's going to depend on how big a project this is and what the research goals are. There's quality control involved, and again, the degree of quality control is going to depend on how sophisticated a biorepository storage operation that you have there. True laboratory information management and quality control includes a 24/7 temperature monitoring system, tracking of the sample, talking about backup freezers and generators for disaster preparedness. And many of us have probably seen news stories or heard about you know, tragedies whereby very important patient samples, you know, even stem cells potentially being used to treat patients in a clinical trial have been lost because of failure of the storage system. And so the quality control and then the backup is crucial to that. I mentioned the LIMS, and of course, all of us on this webinar, we're mostly clinicians. And so the LIMS is a complex IT operation with the biorepository that's well beyond the scope of this webinar, but just important to know that it exists and that it has a very important role in the quality and the standardization of a project such as these. Here's just a list, again, not for us really to create, but for us to know about all these things that are required in terms of disaster preparedness for the samples that we're collecting. You'd hate to take out samples 5 years, 10 years worth of study that we might be doing for a project such as this, only to be losing samples due to disaster. And then finally, Ken mentioned tracking the outcomes. I think it's crucial to have an individual as part of this, whether it's the CRC, a nurse, or a clinical assistant, who's in charge of making sure the patients fill out their patient-reported outcome measures. In my practice, we don't give the patient the procedure until they've completed their questionnaires. And so a lot of times just keeping an iPad or a desktop computer that they can utilize in the room. They can even fill out the surveys and the questionnaires while they're waiting for the blood to process when making PRP. All of that's possible. There's any number of registry options for us to use in research that are out there. I don't think there's any one that's better than the others or that's proven itself such at this point. You definitely want to try and integrate it with the EMR, and a lot of our institutions now have apps that allow patients to engage with both research and their clinical practice through the apps. You got to make sure that it's HIPAA compliant and that it integrates with the biorepository as best as possible. So thank you very much. I'm going to turn it over to JT to talk about this big project we have with the Biologic Association.
Great.
Awesome.
Thank you, Shane. The JT Tokus, as mentioned earlier, orthopedic surgeon at Mayo Clinic in Arizona, past president of the Arthroscopy Association, and helping lead some of this effort, this multi-center initiative with the biospecimen repository. So thanks, JT.
You bet. Hope everybody can hear me okay.
Yep.
Super. Well, thanks very much for the opportunity. It's a pleasure to be with you guys tonight and to talk about this. These are hard questions. If you're going to do good, solid research, then the truth is most of it's just work. And we are blessed to have a great team set up with a bunch of institutions around the country that were willing to come together to do good science and to be willing to donate a bunch of pro bono time to putting this thing together to see if we could get it off the ground. I think most folks thought we would fail. Maybe I may have been one of those at the beginning that was less than positive that we'd get this completely done, but I'm very happy to say that we're on pretty good footing. So tonight, my job is to talk to you a little bit about how we got to where we are and the importance and the sort of justification as well as future directions that we're going with what we call the BARB, or the Biologics Association Registry and Biorepository. My disclosure that I'm an orthopedic surgeon. I'm not a basic scientist. They're available at the AOS in the course book. I have no financial benefits from this or no secondary issues with regard to this particular role as principal investigator of the BARB. I have been on the motion steering committee, which is another large outcomes registry in the U.S. military, which is, I guess, where my experience is. Well, the question is this a problem? And I think this study really highlighted to us way back in 2016 when we started this and showed us that there were over 570 clinics in the U.S. offering direct-to-consumer marketing of, quote, stem cell therapies. We don't even use the term stem cell anymore. Even Arnie Kaplan, shortly before he died, recommended against using the term because they don't actually perform, at least in humans, the way we thought they might perform. And there's a very specific rendered definition. But I will tell you that we remain plagued in our city, as well as you guys, I'm sure, in your cities around the country, where we see a bunch of people direct to consumer marketing, paying cash, and this has unfortunately become a big cash grab. One problem with this is if you take a look at these online direct-to-consumer marketings, advertising, et cetera, consider this article from JBJS back just a few years ago that showed that 95% of websites made at least one statement of misinformation, and that the average was about 4.65 lies per site. So if your clinic is being evaluated by the metric lies per site, I might suggest to you that we probably need to go a different direction to get a little bit more scientific in our approach of how we're offering healthcare to our patients. I'll give you a little personal experience of mine. This is an article that came out of the New York Times a few years ago, henceforth called the New York 12, where 12 patients were hospitalized with severe infections due to a, quote, stem cell strain. 11 patients in Texas, split $1 million, and then the other patient was mine, who was the most seriously injured, and then ended up going to sort of being the chief litigation person in this. I have his permission to discuss the details, although we won't go into it. Roughly, they founded a company. They marketed like crazy that they were doing stem cell stuff. This was out of a chiropractic office in Sholo, Arizona, very small town up north. And they were advertising it, and unfortunately, there were some breakdowns in the sterility process, and a bunch of these patients got really sick. Unfortunately, my patient lived. It was pretty tough. But in response to a bunch of these sorts of events, the FDA came back and said, look, this can't go on like this. We can't have people just going after a cash grab and marketing whatever they want. We are either going to slam this thing home and shut it all down, or you as the physicians have to come up with a responsible way to sort of balance the potential benefits of what these products offer, but also with the responsibility of scientific follow through and accountability that we as physicians know that we need to have. And so out of that, I will say great credit goes to Scott and Connie Chu and a number of people, Bill Maloney from the AAOS, Ken Zaslov, and others that came together back in 2018 and held the first of these big conferences. And there we brought everybody together that was a stakeholder. So this meant surgeons, physicians, people that were supplying it. We brought patients, we brought NIH folks, we brought FDA, we brought regulators, we brought leadership from around the country. And out of this article was published back in 2019 that you may go back and note that we need to get better about the way that we're publishing and the way that we're providing this care. Out of this conference came consensus recommendations from all stakeholders. So again, at the end of this, the goal of this conference was to come together and say, okay, what is our direction forward and how are we going to get to a place where we can promote the responsible use of these products in our patients? Now, here's the key. Biologics are different. They're unique. If you do a joint registry, for example, everybody knows what's in the joint. You have to specifically sort of quantify that to get FDA approval. If you're doing a drug study, you have to work out all of what's in the drug before you can start giving it to patients, pharmacokinetics, complications, all of the specific things that go into that drug that may be bioactive. Well, that's simply not the case in biologics, because we draw some blood or we take some fat or we draw some bone marrow, we spin it at certain times, we do something else, rub dirt on it, chicken bones, whatever it is, and then we stick it in you. And honest to goodness, we have no earthly idea in some respects what's going into the body, and therefore no ability to track it. Hence why it's so important that we come up with these standards. Okay, so we got to develop these, and out of these there were a few that came out. First, we have to define our terminology clearly. We have to clearly design and talk about what we're talking about with clear definitions. So what does PRP mean? Well, you'd be amazed at the amount of discussion and disagreement as to what goes into the definition of PRP, let alone a quote stem cell or these other things. So defining terminology. Second, we got to standardize reporting requirements. So earlier tonight you heard Ken Lovely sort of summarize MIBO and the minimum sort of information for biologics that we need to report so that we make sure that everybody's comparing apples to apples. The third one actually seems obvious, but it took a lot of work, and that was that we needed to get osteoarthritis designated as a quote serious medical condition, and that is a definition that comes through the WHO, but through a bunch of the regulatory acts, without going into too much detail on it. If you could define something as a serious medical condition, it allows us to fast track therapies through the FDA. For those of you that do biologics, you know sometimes the biggest roadblock in your practice is the regulatory side, right? That's the thing that's the hardest thing to do, but it's necessary. We were able to gain that serious medical condition designate. It was not as easy as you might think it would be, but we were able to get that back maybe in 2021. So a big step forward for us, and has allowed us to fast track some of these therapies. Fourthly, we got to clarify a consensus approach for biological markers of interest. What are we going to follow? What should we be looking at? You know, from we talked about platelet count and platelet concentration. Sure, that's the beginning, but there, you know, PRP has over 400 proteins in it. Which of them are important, and how are we going to quantify them, and how are we going to decide how to study them? And I think that requires a consensus approach. Explore these accelerated pathways through the FDA with the approval of new drug applications for biologics to treat these conditions. And then most importantly for my talk tonight is we have to establish the framework for multi-center knee OA clinical trial consortiums. Why this? Well, if you're going to study biologics, you got to start with a pilot. And you should start with something that is common, so knee OA, and something that we do a lot, i.e. PRP. So we wanted it to be as simple as possible, and certainly PRP is certainly not simple. But what we thought was we needed to put a foothold into something we could start with. And whenever you start a pilot project, you learn all the mistakes. And believe me, we made lots of them in terms of how to build this thing. And the idea that we've talked about many times is creating a Lego city. So you're going to start with the Lego center, maybe it's the airport Lego design, and then later you might be able to add the ice cream shop and the fire station and all those other little Lego cities for other aspects. But if you can establish the process and the collaborative, you've got the beginning blueprint, and then we can modify it and take it deeper into other disease processes and other ways of treating these things. So that requires us to establish registries, and unfortunately or fortunately because of biologics, establish a biorepository so that we know what it is that we're putting in folks. So what are unique challenges to biologics outcomes registries? Well, it's an uncontrolled field. The ingredients are unknown. We talked about PRP a little bit. Very troubling to me. Look at some of these studies on PRP alone. You look at PRP and knee OA, most of the results are great. 26 randomized clinical trials, and almost every one of them shows a great benefit. But go to the hip, you got 11 trials, 1,350 patients, no intervention outperforms performed control, and OA was outperformed by saline control over PRP. So completely divergent outcomes in some of these different processes. So that leaves us a few details to consider. Which combination of the 800 proteins in the PRP worked? We have no idea. Did you use leukocyte poor or leukocyte rich PRP? Was your platelet count 2.5x, 10x? What was your dose? How old was the patient that you got the sample from, right? How much volume of PRP did you give? How fast did you spin your centrifuge? What the hell is thrombin activation? All of these things, how does the other guy in town charge 10K for this, and how come I'm not doing that? All these can make your head explode. So you got to be a little careful as we go forward. So a biorepository truly is different. It combines demographics and PRO outcome data, but also biologic analysis of what we're putting in the patient. Not to mention, every patient starts with a biologic milieu. Is your knee swollen? Is the effusion that you have contain a biologic milieu? It's different in your knee than my knee. Well, certainly, but how different? Well, we got to know that. So we've got to take that stuff too. Other registries have strictly defined interventions, but nothing about biologics has been strictly defined so far. So how do we build it? We'll enter the BARB, the Biologics Association Registry and Biorepository. We began with this project to use us to force us to establish standard operating procedures and collective and collaborative infrastructure. You see the institutions that originally got on board down here with HSS, Northwell, Colorado, Cleveland Clinic, Cedars-Sinai, and Mayo Clinic, Arizona, with input from Stanford. And we wanted to start with something, quote, relatively simple, common, and relatively easy. Then we'd build a collaborative infrastructure that could modularly expand to address these existing therapies, and then use real-world evidence as a model, as opposed to a randomized controlled trial. Why real-world evidence? Well, because if you have a lot of it, you can take action on it very quickly, as opposed to a randomized clinical trial, which can take years to do. These are the advantages. It has the power of N to retrospectively see what worked. Literally everyone can be included, provided we can properly analyze their input. Data AI, especially with new machine learning and things like that, allow for cheaper and more modular approaches to the data. And again, it's cheaper and faster, and RCTs only give you information about the one formulation that you picked. Now, why is this important? Well, the 21st Century Cures Act, the FDA did create framework for using real-world evidence like this, like the kind that we are collecting and the registries are collecting, to support approval of drugs and biologics. This is a huge quantum shift and step forward for us, because it will allow us to collect this data and then have actionable data within months to early years as opposed to decades. So the initial BARB trial looks like this: PRP and knee OA. With common problem with a common treatment, we approach it differently. Remember that in a randomized clinical trial, variation is the death knell, but in real world evidence, it's the lifeblood. And that's what we set it up as. We don't want to standardize the PRP. We want everybody to use whatever they use. We want variation. Variation is the key. We just have to standardize knowing what it is. So we don't standardize the PRP, but we must standardize the process and the analysis. Don't care what you put in, but we must know exactly what you put in. Correlate this with a clinical analysis and welcome variation. The nuts and bolts, we have standardization of analysis at all sites of blood volume and cell count. Again, these vary depending on what you put in the knee, but we know for each patient what that blood volume and cell count was. We know what the analysis of PRP is, thanks to the HORIBA that Ken mentioned earlier, cell count, platelet count, volume, platelet dosing. We all standardized our PROs, patient-reported outcomes, with the KOOS, the knee osteoarthritis score, being our anchor score, but with which we ask a lot more: Tegner's, Lysholm's, pain scores, etc. X-ray classification for KL grade, and then we inject as normal. You just do your normal standard of care, with the exception that we save 1 cc from all PRP, and when we store those in a standardized fashion. Now, you have to store them locally, because if you transport them, it affects the concentration. If you shake them too much, it affects that. So everybody has to store locally with a local biorepository, but then you have to centrally analyze them, because that will induce too much variation. So we're going to send those to a central lab for the ELISA analysis, and then we'll correlate those to a biologic environment. Okay, so that includes 6 sites. We're powered for 1,200 patients, over 45, actually, we're up to 60 growth factors. And the PRP pilot has built the engine. In terms of SOPs, there's clinical infrastructure that required collaborative IRBs. It requires data sharing and IP agreements. We had to go to FDA for IND and IDE permission. We had to get key personnel involved. We have REDCap as our chief outcome score. And of course, then we look for funding opportunities. And the concept, again, is this. Ultimately, it's a Lego set framework where we can build on as we get better, ask new questions, find new pathologies, and find new technologies that might be able to approach this. So the next question, of course, is just great. Everybody's really interested. You can't believe the number of thumbs up emojis that Ken Zaslov and Scott and Shane and I have seen. Man, guys, we think this is a great idea. Well, who's in for funding? Everybody. Everybody was interested until industry realized, hey, how will this get us a FDA clearance on our formulation? The answer to that is no. Why? Because we're not using specifically your formulation. The funders are government funding. Which form of PRP will you use? We said, well, we don't have one. We're not testing a single product. We're testing all of them. But then they said, well, you don't even have a hypothesis. We're like, right. Our hypothesis is, let's figure out what the correlation between outcome and response is and what we're putting into patients. And that doesn't fit very well with many of the major funders. So that's been pretty tricky in terms of getting funding over time. So the grand ambition was, you go back to the groups and you say, are you guys willing to put this involved in your own institutions? And I will say very proudly, every single institution stepped up. And that required a tremendous sacrifice on the part of each of the institutions. Had to agree on a protocol, SOPs, standardized testing equipment at the bedside, get your IRBs approved, get data sharing agreements, get internal funding, collect the PROs rather, store locally, and then batch ship. All a ton of work. And of course, remember, we start this in 2020, 2019. And so here we are in 2024, and many of the institutions are just getting up and running. But I'm proud to tell you that, and from a status update, I will say as of May 25, all sites were up and running. And as of last week, we have actually 746 of our initial 1,200 required. So well on our way past halfway. We have SOPs that are finalized for most steps, including our data sharing and material transfer agreements. Those were executed back in May. And then updates just to share you with in the last minute here. The AAOS and the BARB have agreed to partner. The AAOS has decided to bring the biologics, the registry, under the AAOS family of registries, combine that with the BARB's work, and call it the Orthopedic Biologics Registry, or the OBR, if you hear us talk about that tonight. The AAOS establishes PRO registry framework, leveraging their skill in orthopedic registries, and the BARB continues to do the biorepository work that includes sample collection, storage, shipment, and analysis. The two will then combine the biologic and the clinical data in a third-party framework called the OBR. And then ANA, AOSSM, and the BA will officially adopt the OBR as our official registries. And we have tentative agreements from all involved. In 2025, we obtained our first federal funding to establish Walter Reed Army Medical Center as the first military partner in the biorepository. 2026, we have got now permission and funding to expand from the original 6 up to 10. And then in terms of future directions, we need to successfully partner with the AOS to build the OBR. We eventually hope we'll get to 10 qualified institutions. These are institutions, we're not overly picky about it. They just have to have a decent number, and that they're willing to follow the SOPs, and they have the infrastructure necessary to be able to do the scientific work that is necessary. We plan to use pilot data to obtain programmatic funding. We plan to expand to other therapies and other diseases. We have another study that's starting off in the field of exosomes to test that regard. And so we've built the engine, and now we hope to allow the science to separate the magic from the mischief that's clearly out there. And like any worthy journey, mostly it's been painful, but well worth the climb. And it's been a tremendous collaborative so far, with a ton of work to do and a ton of ways to go. So with that, I'll stop and thank you very much for your attention.
Great summaries. Great overview. Thank you all. That really sets the stage. We have a little time for some brief discussion. Yeah, you're right, JT. It has been it's painful. It's been challenging, but it's worthwhile. Nothing hard is ever easy. And because this area has such tremendous potential, but so many unknowns, we just need this. I mean, we need a registry for anything we do in orthopedics. I submit to you almost nothing more than regenerative medicine, just because of the heterogeneity that we've all pointed out here. So, Ken, you talked about the MEBO criteria. So, but to this issue of collecting data, this minimum information for biologics and orthopedics, there's a whole, laundry list of things you should collect. Do you do that? I mean, what is practical? How do you practically do this? How much of that do you routinely collect in your institution?
Well, I think, all the things in the first 6 or 7 slides I show you, we collect every time we do PRP, or BMAC at this time. We don't do all of the MEBOs, but of course, we're not really doing stem cell research yet. When we do basic science research at Feinstein, with our lab with Dan Grandy, we're looking more at the cell characteristics and purity, and we label all of that and we keep track of it. But these are all in animal studies. In the humans, we mostly are doing the first 6 or 7 slides I showed. And of course, at the end of this, if Steve wants to get our slides, they're perfectly welcome to pass them out to any of the participants who want to have a little more data and didn't have time to take notes when we spoke so quickly.
Yeah, I think it's a good point. I think there's criteria. There's a lot of, many of them are important. Some of them are a little more esoteric. At the end of the day, our journals need to require some basic information to characterize what we're putting into patients. Whether we follow the MEBO criteria to the letter, I think the spirit of the law is important, that we really characterize what is, so we can compare studies, and the studies can be reproduced. Shane, great overview on all the lab stuff that we can do. And so as you watch your slide, it can be complex, having all the laboratory resources and freezers and backup systems, all those things. What are the simple things that are practical for an individual who wants to contribute to this area but may not have access to, a very involved, well-stocked laboratory? What are the simple places to start? Is it a simple CBC?
Yeah, I think it is. When we first started doing this 15 to 20 years ago, we wanted to do those quality controls, and we wanted to make sure that the products that we were putting into patients met the definition of platelet-rich plasma, and we weren't just injecting with whole blood. So I think it's a simple matter of partnering with your core lab that you would normally use. You can do a simple CBC. There's some calculations that you have to do to account for the fact that this isn't whole blood and that it is PRP. And a lot of times our laboratory colleagues can help us do that very simple calculation. So we have done that in the past. And then, there are some success stories out there. Don Buford has a well-established program in Dallas, and he sends out a lot of his labs and has been very successful in tracking his outcomes and getting a laboratory printout for the before and after of PRP or BMAC that he makes. So it can be done, and you can even store samples very simply to conduct simple waste-related research. If you're looking for a small number of biomarkers, let's just say a multiplex of 25, and you can get a simple low-risk IRB and a -24 freezer to keep some samples in and bank them till you have 25 and run those on a multiplex. It sounds complicated, but it is just sort of basic laboratory techniques that we could all participate in.
Scott, I would just add one thing, and that is, so for the people who are listening, let's say they want to do something on simple like BMAC or PRP for rotator cuff. I just think if we were, even if you're not going to have a biorepository and be able to look at all the proteins and what's in the drug at the details that we're doing, if you're going to report on success rates, at the minimum, you have to have platelet dose and white cell characteristics and volume. I mean, I think, even if you want to do the simplest thing, you really can't report just saying, as we know that there was two big reports in Australian literature showing that PRP is no good, but both of those used machines which were only concentrating 1.6 to 2x. And that's very different than what we're doing now with anywhere from 5 to 10x. So I think that, those simple characteristics, how many billion platelets, how many, what was your white cell type, how did you draw it? Those are simple things that anybody can do if you're going to try to report on the outcome of a straightforward injection procedure.
Yeah, good. I'm glad you mentioned white cell types, not just white cell count and leukocyte number. It is the differential, and that's rarely reported, which is, kind of surprising. We may want to eliminate neutrophils, but concentrate other macrophage subpopulations or T regulatory cells. So it's, PRP is very complex, and so starting with the CBC makes sense. And to that point, Ken, or you or anyone, JT, are you doing CBCs in your routine clinical laboratory? We all have the Cerebra machine. We can do it in our own labs, but that's not practical for most of our colleagues. Are you using your routine clinical laboratory?
We are not, and I think the main reason is because the laboratories are running thousands and they have all these filters. And my concern is they see a PRP. If you do the CBC on a PRP, that platelet number would scare the heck out of anybody who thought it might be a blood sample, and there'd be all kinds of alarms going off. So we don't use the central lab. You could do it if you could work out some very specific protocols and separate it so it doesn't go in the patient's chart and freak out the oncologist or somebody else if this patient has some disease.
Yeah, ultimately, I agree. It'd be good to get to the point where we can use our clinical laboratories. Now, of course, the other question is, how do you pay for it? It's not standard of care. It can't really be billed to insurance. It's
so that's our big problem too. So we're lucky enough that we have Horibas at each of the sites that we do this. So 3 different Horibas at our main sites, and so all of our PRPs, at least at Mario, Arizona, right now, are all done by Horiba, and we don't have to send them to the central lab. For exactly your point, Scott, there's a number of things. One is that then of course you got to bill the time and the effort and the readings of it, right? And so who gets to bill that? And then is it part of a research protocol? Blah blah blah. It increases the complexity and the cost. But the other thing is we compared CBCs and platelet counts at our lab versus on the Horibas, and we found that the transports themselves change the concentrations wildly, because sometimes they sit in the office for 6 hours. Sometimes they get shaken. Sometimes, especially here in Arizona, they get hot. So we now know that you can't trust a laboratory to, at least this, you can trust our laboratories, they're amazing, but the sample that you take out of that patient is not the same concentration as the sample that ends up in that lab. That's why we believe, and why one of the SOPs for the BARB is that you must have a Horiba at the bedside and check those counts right there and right then when it comes out.
And that is a very important point. As you mentioned different sites, and I was going to ask that exact question. What are the challenges of transporting a specimen from a satellite to a major center? So, your average individual may not have a Horiba, may not be able to do it point of care, but they want to participate. They have a lab they can use, but it's cross town. Or what, and you kind of touched this already. What are the challenges of transporting specimens in a timely fashion? And how do we standardize that? And maybe it's, it's a rhetorical, it's a question we don't have all the answers to.
Really tricky.
Recognize the challenge.
But Shane talked earlier about the importance of the clinical research coordinator. This is where it is, if you're the provider who's doing these things as part of your clinical practice, I'm not going to say it's impossible, but I bet you it's impossible. So you have to invest in that infrastructure of those people, that team. We're blessed here at our place. to have a team that is all invested about how to make sure that the sample gets taken and frozen so it's not lost in translation, and that those numbers get recorded and placed in the record. Because if that's left up to me and maybe up to some of you, the data is going to be so full of holes it's going to be unusable later. So it's a big commitment, and it is absolutely a team sport. And finally, of course, the freezers, right? The biorepositories that we're storing these in. It isn't just a freezer, because how do you know that the temperature has stayed solid, et cetera? And sometimes these are going to be stored for months and years. So yeah, it's a team sport, and it's a big lift at the front end. And then hopefully if you've got it and get everybody involved, then it becomes a little bit more autopilot-y later.
And to that point about storing specimens, so right now we've been talking about CBCs, and that's a standard place to start. But there are admittedly so many other assays that we can potentially do. Ideally, we need to identify what are the sentinel markers of purity, potency, biological activity. Maybe we need to extract RNA for transcriptional profiling. Maybe we need to process for proteomics. Maybe we need to use it in cell culture studies. I mean, the point being, there's a lot of different assays we may eventually need to use, and that would determine how we process and store. Room temperature, -80, -196, all those questions. You mentioned impossible, and I agree as far as doing it ourselves. Speaking of
That's
impossible,
one of the reasons I think, like JC said, this real world data, we hope that this pilot helps us. We'll sort out, okay, where did we find some things that seem to make a difference? And then for the bigger study in the future, we would hopefully be able to aim for those particular markers, whatever that might be. But right now we have to figure out where is the beef.
Yep. Well, and the last question I was going to ask the group, and Shane, it is the challenge. Talk about challenges. Patient compliance with PROMs is so challenging. Shane, you made an interesting point that I think you said patients can't be seen at your institution until they fill this out, right? And Cleveland Clinic, I know, has a system in place similar. We're trying to get there at HSS. We're not there, but just your last parting thoughts on the challenges with patient compliance and the responder burden. Ken, you mentioned you can't have 10 different PROMs, for patients.
Yeah, I think it really comes down to a little bit of hand holding and personal touch when it comes to this. So when I consent the patient, I first thank them for participating in the research study, and then I tell them how important it is that beyond the procedure that they're going to get, that they help us grow this practice of orthobiologics by helping advance the science, and that the only way we can get there is by doing these questionnaires. And so, yes, that first questionnaire, the baseline questionnaire, must be completed either before they show up for the appointment or while the PRP is spinning. They do not get the product until that is completed. And then beyond that, it comes down to that staff with continued communication every time we see them back, how important it is, and we thank them for doing it, and then making sure that my clinical assistant or my nursing staff regularly contacts the patients. And when we've done it that way, we've had tremendous compliance, at least out to about 2 years or so. It's worked really well.
Right. And we can learn from your experience. I think it's important. It does come down to human resources, ultimately research assistants that can help follow up with the patients. It's challenging. So, well, with that, I think we're right at about an hour, and maybe we should wrap it up. So I thank you all for it.
I actually had a question. I'm actually designing a trial right now for adult recon guy here in Florida with PRPs. And one question we had, which we had a little bit of a pushback, what is the legal framework for this when it comes to IRB? Does it differ in any way with any other regular retrospective study or a prospective study? I think Shane, you touched base to it a little bit, but is there any difference, especially when it comes to keeping a sample?
Yeah, it's really a question of consent, so you just have to make sure that the patient consents to it. Because it's autologous, it makes it fairly easy, because, giving patients their own stuff, if you will, is a little bit of an easier regulatory trial. It does require that consent form, but from a regulatory standpoint, beyond that, storing it, just have to make sure that they can withdraw at any time with no penalty and blah, blah, blah. And then you have to have protocols in place about what you're going to do with the specimen should they decide to pull out. Then there's another layer if we're going to combine data and ship specimens to a centralized place, which you're going to need to do if you have a big multi-center place. If you're going to analyze it outside your institution, that requires another process, but it's not undoable.
Okay.
Last point I make, really, this probably doesn't relate to just PRPs, Peter, but if
Mhm.
on any of these samples you plan to do genetic testing, that's a separate, it's a different entire consent form, or it needs to be stated in the consent form. At our institution, that's a separate paragraph in the consent form. So just keep that in mind if you're banking specimens for later downstream analyses where genetic testing may be carried out. And then at the end of the day, simple transcriptional profiling and gene expression analysis would probably
Mhm.
fall in that area. So.
And one other question, you mentioned the CBC. How do you usually handle the payment for it?
The payment for what? Sorry.
For the panel.
We can't charge the patient because it's not standard of care.
No, we don't charge them for the test.
We pay
do
for it out of
that's our
research
own-
funds.
That's our own cost.
Same.
That's an important point. You can't charge a patient for something that's not yet standard of care. Maybe in future will be, but the point is we don't get the CBC back until well after the patient is dosed. So we're not using it for clinical
Mhm.
decision making. We'd like to get to that point maybe someday, but unfortunately we're not.
Okay. Well, thank you very much. I learned a lot. It was very, very.
Thanks a lot
Oh,
for
yeah,
inviting us. It's
Learn
nice
a
to
lot
see
from my
you.
colleagues too.
Thanks for having us.
All right, gentlemen, we'll sign off then. Thanks so much.
Thank
Thank
you.
you
Thanks,
again to
Scott.
Smith and Nephew for their support. That helps us get this done. Thank you.
Thank you, guys.
Bye-bye.
Good night.